that you will follow us on social media. We're on Instagram, Facebook, and Twitter. And you can even follow and subscribe to our YouTube channel. So we hope you do, so you don't miss any of these ALKtALKs. And we're so thankful for Mark for putting this on, and for Dr. Awad and Dr. Chiarle for coming on a Sunday night. And I can't wait for you to hear more about our shop from our very amazing member of our group, Erica Lavacek. So I'm going to hand it over to Erica. Go ahead.
Thank you, Gina. Okay, hi everyone. I'm Erica Lavacek. I've been a member of ALK Positive since 2017 when I was diagnosed. Currently on lorlatinib, 15 months and counting. I'm also on the board for ALK Positive Charity and run the ALK Positive store. And that's what I want to talk to you briefly about today. So thanks to everyone who has already purchased an item. We've had several items purchased. A percent goes back to the charity of each order, and you can access the store on the charity's website by going to ALKpositive.org and click on store, and then you can see everything that we offer. We have several items with the ALK Positive logo, and recently we just added several new additions that are based all around research, what we are all about. We have designs with the molecules, compounds of alectinib and lorlatinib. We have team research shirts, and we have a couple choices for more research, more life shirts. So please check out our store and visit it. And speaking of research, I cannot wait to hear our talk about research. Thanks everyone for listening.
Okay, Callan, please, in the chat, you can enter your question to everyone in the chat, and I'm going to turn it over to Callan and Emily.
Terrific. Thank you, Mark. So yeah, it's a great line of clothing that Eric has set up for us, and I hope that everyone will go to www.ALKpositive.org and purchase something, and it's very comfortable, high quality clothing. Dr. Mark Awad is one of our longest running researchers with ALK Positive. He received an award for starting the translational part of the vaccine research in 2018. I had the great pleasure of meeting Dr. Awad in Santa Monica in 2019. And I've never met Dr. Chiarle in person, and I've probably mispronounced his name, but he'll correct me. And but I have spoken with him, and anyway, they're both wonderful, wonderful people. Dr. Awad has taken over management of the spacewalk project that I think a lot of you are aware of, and he'll be talking about that and answering questions about that. Obviously, a lot of you have questions about the vaccine that he's developing, and we'll be talking about that. But he's also a great clinician. I've heard nothing but great things about, you know, just his personality and his knowledge with ALK positive patients and many other types of lung cancer as well. And you can ask clinical questions. Now, what will happen is that he won't specifically answer to your case, but he can answer sort of generalizations. So please feel free to put into the chat clinical type questions as well. He's extremely knowledgeable. And I'd ask you to not put into the chat sort of just informal conversations between one another, because Emily Venanzi, who's a member of the medical committee, will be helping me read through the questions. And it makes it really hard if we have to read through all sorts of things saying, "Hi Peter, how you doing?" So just keep it to questions that you want to ask Dr. Awad and Dr. Chiarle, please. So with that, we're going to start looking at the chats. And while you start entering questions, I'll open up with a couple of questions. So Mark and Roberto, if you'd like to tell us a little bit about yourselves, that'd be fabulous. How you got into oncology, how you got into research, how you got into the specific field of lung cancer for oncology, and how you got into the field of developing vaccines, and just kind of get us rolling on that for a little while. That'd be fabulous.
Great. Well, maybe I can go first and then turn it over to Roberto. So thank you so much for the invitation to participate tonight. And thank you to Colin, Gina, Erica, Mark, Emily, for everyone organizing these wonderful talks and events. I've heard amazing things about these Sunday night chats, and I'm really happy to be here. So I see also some familiar friends and faces and names across the Zoom board tonight. So hello to everyone out there. I hope everyone has been safe and healthy. I know there's a lot of challenging times people are facing, both from an oncology standpoint, but just also things in terms of the weather and in different parts of the country. So I hope everyone is safe and warm and healthy tonight. So as Colin mentioned, my name is Mark Awad. I'm the clinical director for our thoracic oncology group here at Dana-Farber Cancer Institute in Boston. So I did my residency. I went to medical school at Johns Hopkins, did my residency at Mass General, and my fellowship in the program here in Boston, which is between Mass General and Dana-Farber, and I've been on the faculty at Dana-Farber for about the past 6 years or so. Maybe to your question as to how I first got into oncology and what maybe directed me towards some of the fields that I work in. So when I was back in college several years ago, my father was diagnosed with a metastatic cancer, a different type of cancer, not lung cancer, but a cancer called GIST, which stands for gastrointestinal stromal tumor. When he was diagnosed, it was called something else at the time. And I was a college student. I wasn't in medicine yet. I didn't know anything about cancer or about his type of cancer. But it's a rare cancer. And I know, when we talk about ALK, it is not the most common type of lung cancer, although it affects thousands and thousands of people. And so I think to be able to get information quickly about a less common type of cancer is challenging for many of our patients, and certainly was challenging back then when my father was first diagnosed with his type of cancer. This was, when there wasn't a lot of information available in the internet. There were very few patient advocacy groups. There were very few patients who were able to connect with one another to spread information. So it was challenging, to say the least, as a family member to get information. And at that time, since this was a relatively understudied type of cancer, there weren't many effective therapies. So I remember we grew up in New Jersey, and so my father had initially a surgery at his local hospital to remove the cancer, but unfortunately his cancer kept coming back and back. He sought other opinions, and fortunately we were only an hour outside of New York, and went to Sloan Kettering to get some additional opinions. So he had participated on a number of trials, had additional surgeries, had various types of chemotherapy, but unfortunately we as a family kept facing the challenges of recurrence. And at that time, there was just some information coming out that there was an abnormal kinase or an abnormal gene that was responsible for driving his type of tumor. This is a gene kinase, kind of similar to what we see in patients with ALK positive cancer or other types of lung cancer. And so he enrolled onto a clinical trial with an inhibitor of a gene called KIT, K-I-T. And amazingly, after starting this pill, which he started, it was not an FDA approved pill, it was a bunch of letters and numbers, but his cancer started shrinking immediately. And it was the one therapy that seemed to work. And so he has been on it for 15 years now, and is doing amazingly well. And so I think, that showed me the promise of cancer research. And through an understanding of biology, can we develop improved therapies? And when I think back over the 15 years and all of the ways in which I've benefited from his fatherly advice and counsel, and all the life events that he's been to because of research, I think, that certainly helped to fuel my drive and interest in becoming an oncologist myself, and then also trying to work towards oncology research to develop new therapies for patients. When I was a fellow, I worked under the mentorship of Dr. Alice Shaw and Jeff Engelman at Mass General, and I saw firsthand about what wonderful physicians they were, how dedicated they were, but also how they were using their experiences and their talents to try to bring improved therapies to patients. And so that's, I think, what has driven me towards studying lung cancer and to developing therapies, improved therapies and immune therapies specifically for patients with lung cancer. So I'll stop there. I was delighted to also meet Roberto Chiarle when I started on the faculty at Dana-Farber, and he and I have been close collaborators over the past several years and have been bringing some of his basic and laboratory research into the clinical space to try to launch our vaccine clinical trial. And so Roberto, I'll turn it over to you to introduce yourself and share your background and how you got into ALK and vaccine research as well.
Yeah. Thank you, Mark. Hi to everybody. It's a pleasure to be here on a Sunday night. It's I think the best use we can do for a Sunday night instead of watching TV or doing something else. So I really like the idea of doing that. And thank you, Colin, for the invitation. Of course, Gina, Erica, and Mark for organizing all of this beautiful event. And much less than Mark, I recognize few names in the audience. So it's nice to see you virtually. So my last name is so difficult to pronounce because originally I'm from Italy. So Colin, you did quite well. Mark is a step ahead because it says Chiarle instead of Chiarle, which took 2 years for Mark to learn how to say it, but now it's very good. And so I'm originally from Italy. I'm a pathologist, so still a clinician, but not that type of clinician that directly sees patients. I more work on a microscope that is on my back, so I stay many hours sitting there on the microscope, looking at lung tissues and other type of tumors, right? Trying to understand through a lens what could be the driver force behind tumors, and of course trying to find some therapy. And I moved to Boston in 2012, so it will be next year will be basically a decade. And the reason why I came to Boston actually is, well, not only because, of course, the urban environment is very attractive, but because I first developed this idea or trying to tackle or target ALK-driven tumor in Torino when I was a professor there, and I'm still a professor in the University of Torino, by the way. Torino is in the northwest of Italy. Maybe most of you don't know this name. It's not one of those famous Italian cities, but still is a quite nice one. And so I was there. Of course, I did my fellowship in New York many years ago. Well, not so many, but a bunch of years ago. And then I went back to Italy, and I basically became a professor over there. However, and I started to develop these ideas, and then I felt that I needed a bigger arena really to try to move this concept to the clinical world. And I remember that when we first thought about developing a cancer vaccine against ALK, of course, we were working in mouse models because we couldn't do in patient. Hopefully we will do soon in patient now. But now that everybody is familiar with vaccine because of COVID, everybody knows that there are several types of vaccine based on DNA, based on RNA, based on protein, based on peptides. So now everybody is an expert of vaccine. And likewise, for ALK vaccine at the time, we were using a DNA-based vaccine that, however, in contrast to COVID, never really worked for cancer. I mean, DNA-based vaccine for cancer don't really work well. So there is need for a different formulation for more sophisticated formulation like peptide based or protein based. And I didn't have the possibility to do that in Italy. And of course, there was not the environment that was supportive for such a complex development. So this is one of the reasons why I basically accepted the offer to come to Boston in 2012. And there, I remember that I went after, of course, when I arrived in Boston, I tried to communicate with clinicians about the possibility that the vaccine could work in ALK positive lung cancer. However, the clinicians were quite dismissive because they would say, no, Roberto, there are now beautiful ALK inhibitors. That will be the final solution for ALK positive lung cancer, ALK inhibitors, which is true. I mean, as we saw by the poll, most of you are on an ALK inhibitor and they work amazingly well. However, we realized after a few years that that's not the final answer. It's a fantastic answer, but not really the final one. So it took a few years to build again their interest for immunotherapy for ALK positive lung cancer. And actually, I went to the director of the thoracic oncology, the Dana-Farber at the time, to try to move this forward. And he told me, Roberto, you need one single very bright, promising, new young investigator to bring this one into the clinic. Otherwise, it will never work. And so he gave Mark Awad name, and that's where we started our contact, right, a few years ago, because he was indicated to be the brightest new young PI at the Dana-Farber for ALK positive lung cancer. So that we started to connect, and to credit of Mark, he immediately realized that the ALK inhibitor were not the final solution. So he immediately understood that immunotherapy could be a complementary way to further improve the efficacy. And we started to collaborate, and then we wrote the first longevity ALK positive grant that eventually was small at the beginning and that grew, and here we are. So I can stop here, and so we can move forward to the next step, but this is basically the brief history or how we came to this concept.
You both had wonderful stories there. And Mark, I'm so glad to hear that your father is still going really well after 15 years on that inhibitor. That's absolutely an amazing story and very inspiring to all of us. Roberto, we're so pleased that you made the trip from Italy and you're here with Dr. Awad now. So I'll pose a question. There's hundreds of different types of cancers. Why did you choose ALK as the type of cancer that you would focus on for vaccines?
Mark, you can give your clinical perspective, then I can give my research perspective of that. Yeah.
I think it's a great question, Colin, and I think it kind of there's several reasons. I think it gets down to the fact that there are some mutant cancer proteins that also have normal functions in the body, or normal versions of expressed proteins. For example, some of you may have heard of EGFR mutant lung cancer, and in the body there's a normal EGFR protein that is involved in normal cell functions, and then the mutant form of the protein is what causes that version of lung cancer. And so that makes it quite tricky to attack cells that make that EGFR protein, because it runs the risk that in addition to attacking cancer cells, you may also be attacking some normal human cells where that protein is found. I think what is potentially very, very promising and unique about ALK, as well as a few other subtypes of lung cancer or other cancer types, is that the ALK protein is virtually not expressed in any normal adult lung cells. And it's unique in that the cancer cells are typically the only cells in the body that make this protein. So it hopefully means that the body, isn't used to having the ALK protein. It's not what we call tolerant to the ALK protein as much as it's tolerant of other types of cancer proteins. And that because there's such a unique differential where the ALK protein is in cancer cells but not in normal cells, that really hopefully gives us the ability to harness the immune system to attack ALK and thereby hopefully only attack cancer cells without attacking or normal cells in the body, which runs the risk of potential side effects. In addition, I think, and I'll ask Roberto how he started working on ALK and how he developed his ALK vaccine. But I think also having such a close and inspiring laboratory collaborator as Roberto, I think his work really has demonstrated with a great deal of evidence in mouse models of ALK positive lung cancer, which there are differences, of course, between mice and humans, but other therapies have moved into clinic with less evidence, and so I think with all of the really wonderful supporting evidence that Roberto's efforts and research have generated, I think that has also really inspired me that this could be a promising and hopefully effective therapy for patients. And so Roberto, maybe you want to.
Yeah, Mark, you are perfect. I would just add one more point. So this differential expression that exists between ALK positive tumor that express quite a bit amount of ALK, of course, which is the target of the ALK inhibitor, in contrast to normal tissue that do not express basically ALK in any tissue, was a key point, because in principle, vaccine could be generated against, I wouldn't say any protein in the body, but many of them. But you want to avoid that the vaccine will recognize normal cells to generate off-target effects and side effects that are unpredictable. So this was one of the reasons, as Mark pointed out. The second important reason is that it became clear in the field of ALK positive tumor in general, because you know that ALK is not only found in lung cancer, but also in other tumor types, including lymphoma, neuroblastoma, and other type of tumors. And lymphoma, ALK positive lymphoma, were studied a little bit more than lung cancer at the time, because as the name says, ALK stands for anaplastic lymphoma kinase. So it was a gene, a kinase discovered in lymphoma first, eventually was then discovered in lung cancer as well, but the name ALK is for lymphoma. And so the field of lymphoma was basically, I would say, at least 10 years ahead, because ALK was discovered in lymphoma in 1994, whereas the first discovery in lung cancer was in 2007, so 13 years later. And so there was 13 years of time to study what ALK was doing in lymphoma, and was clearly evident that patients with ALK lymphoma were spontaneously reactive to this ALK protein, and they were spontaneously generated an immune response against the protein. Immune response means antibody, exactly like COVID antibody, once again, is a good parallel, but also an immune response mediated by cells, lymphocytes in particular, typically T lymphocytes. And so it was very clear that because this protein is not expressed in normal tissue, when it's expressed by the tumor, the immune system reacts. And this activation of the immune system was a very good sign, implying that the protein was seen by the immune system as some sort of novel protein expressed by the tumor cells. So the idea of the vaccination was basically to use this spontaneous immunity that exists in patients. It's very likely that a good subset of you has this type of spontaneous immunity, and try to boost through a vaccination to a level that will generate some killing activity against the tumor. So that was the idea. Take something that is naturally existing and just bring it up, potentiate as much as we can to finally get some activity against the tumor. So that was one additional point why we think this vaccination for ALK could be hopefully a good idea to work.
Terrific. And tell us a little bit about developing a vaccine. I mean, if you go to Chiarle, do you think that's the final version of the vaccine, or do you think it's something that's going to evolve and improve over time? Kind of just tell us a little bit about how it got to where it was when you started the translational mouse models, and whether it's evolved even further since then, and whether you think there'll be even further development. Just kind of tell us a little bit about how that all works, and how's a vaccine made, for that matter.
Great. So I see also a lot of really astute and wonderful questions coming through the chat as well with regards to the vaccine. And so I'll mention a bit to start and turn it over to Roberto. So, some people ask, why is this called a vaccine? I thought a vaccine meant it prevents a disease from happening. So, such as the COVID vaccine, it's meant for people that don't have the infection to prevent infection. And I think right here we're using it a little bit differently, where even among patients who already have an established ALK positive cancer, the point of this vaccine is to use small pieces of the ALK protein as the vaccine product. So these are peptide or protein-based rather than RNA-based, like the Moderna and Pfizer vaccines. These are little pieces of ALK protein fragments that are injected under the skin, similar to like a flu shot or the COVID vaccine, if anyone has had it within the group here. And the hope is that by kind of flooding the immune system with a large quantity of those pieces of the ALK protein, that you will generate an immune response to the vaccine, and then those immune cells that are stimulated against ALK will then also circulate throughout the rest of the body and seek out the ALK pieces wherever they are to attack those cells. So the vaccine, and I'm sifting through some of the questions here as we're talking. So how will we know if a vaccine is working in patients? And that's a great question. So to run a trial, requires funding, and gratefully we've received wonderful funding through the ALK Positive community, the Longevity Foundation, as well as a few other grant sources. But the funds initially will be for about 24 patients. So it's a relatively small number, but we hope that promising results seen from the initial patients on the trial will fuel additional investments, but additional enrollment to be able to enroll many, many more patients. So the fastest way to tell or to convince people that any treatment is working, a vaccine, a new tyrosine kinase inhibitor, et cetera, is to start with somebody who has measurable disease, and particularly if the cancer has been growing on the last therapy. If you switch to a new therapy and the cancer starts shrinking again, I think that is very, very convincing to say that this is a potent and effective therapy. Some people have asked in the chat or elsewhere, if I'm having a great response to whatever I'm on now, like alectinib or latininib, et cetera, why don't I add the vaccine to that in the hopes that the two therapies will be working in concert together to fight the cancer in different approaches or through different strategies. And I think ultimately that will be a great goal for us to have. The challenge by starting with that approach, however, is the ALK inhibitors can work so well on their own initially and for quite a long time as well, which is great. We need to do better, but they can work for years alone. And so if you add a vaccine to alectinib, so say for somebody who's just started on alectinib a few months ago, it will be very difficult to disentangle what's going on. Is the long-term benefit seen because of the vaccine, or might you have hopefully seen that anyways with alectinib or lorlatinib or something else? So for that reason, the initial trial will be enrolling patients who have had disease progression, meaning growth of the cancer on the prior ALK inhibitor or prior therapy. And for some patients, they'll be able to continue on whatever ALK inhibitor they've been on, and the vaccine would be added onto that in the hopes that we will then induce a response. And so we will be looking primarily with scans, but we will also be measuring things like circulating tumor DNA, and hopefully we'll see things that were increasing. We'll hopefully see that that starts to decrease as well. We will be measuring immune cells in the blood, and hopefully in response to the vaccination series, we will see the generation of what we call anti-ALK T cells with the vaccines, vaccine administration. So that is the kind of general principle about the vaccine. Now, the immune system is complicated and tricky, and everybody's immune system is different, which brings up the topic of what we call HLA typing. So many of you know, you can't necessarily take blood from one person and transfuse it to any other person, or you can't do an organ transplant from one person and give it to any other person. You need to have matching for these types of approaches, because everybody's immune system is different and would handle exposure to different proteins differently. And so we are also learning this about ALK, that depending on each person's immune system, one person might chop up the ALK protein and present it to the immune system differently from another person, differently from a third person, fourth person. And the way that we discuss that, the way that these ALK peptides are sort of chopped up and displayed to the immune system is through what's called the MHC molecules, major histocompatibility complex, which is indicated by someone's HLA type. So we've been doing HLA typing among the patients we've seen with ALK positive lung cancer to kind of understand how people's immune systems handle ALK. And we've been trying to develop these peptide pieces of the ALK protein to match different people's immune systems. So the initial trial will be HLA matched for a couple of immune types, with the hope that over time we'll be able to expand to more and more rare immune types. So Roberto, maybe I can ask you to add a bit to that.
Yeah. So yes, that was great description, Mark. I need to make a comment because I'm reading through the chat with all these questions, and I'm so surprised about the quality of the question and the competence of the audience. I mean, the question that you are asking are excellent and are exactly the same question that we get from top scientists. So all my compliments to your insights that are extremely well taken. And there are so many questions. So we hope we will be able to answer all of them, but if not, maybe you can retype again, insisting. I want to know the answer. So let's continue and end up a little bit on top of what Mark just said. One of the reasons why the mouse vaccine does not work in human, as was one of the questions, is because the mouse immune system is different from the human system. So all the discovery that we made in mouse, which took many years of work to identify the portion of ALK that are immunogenic in the mouse, to have a meaningful model that we could use to predict the ALK vaccine efficacy, unfortunately are completely useless in human, because the human immune system is completely different. So we needed to start back from scratch, and that's one of the things that I did here in Boston, because in Italy it was impossible. So we needed to re-identify all these small fragments of protein that Mark was saying about, specifically for for the human immune system. And we were successful in doing so in this HLA haplotype that Mark were mentioning, that basically are present in about half of the population. Of course, our effort currently is to expand the potential application of the vaccine to the entire population. And so we need to do more discovery. So we already started in collaboration with one of the top group in the world, I would say, that use sophisticated technique, because you need to think that you need to identify this small fragment of proteins on the top of the surface of the cancer cells. And that's not easy, but there are very sophisticated techniques that are called mass spectrometry or similar that allow to do that. And so now we are trying to identify ALK peptide also in other HLA haplotypes, so that we could eventually in the future provide the vaccination for almost the entire population if we are lucky to identify them. So that's one of the explanation. And that's also answer that the current formulation is based on peptides. However, given the success of mRNA, RNA vaccine in COVID in particular, because you have to remember that COVID was the very first RNA vaccine approved after 10 years. There is this nice history, you could read that about Moderna. Moderna was founded here in Boston 10 years ago, and for 10 years was struggling to have anything working with RNA vaccine against tumor or other things. Basically, nothing really worked until COVID came, and they successfully developed a COVID vaccine, and now Moderna is really skyrocketing, right? But it took 10 years, and for them, COVID was a lucky event, if you would want to say so. I don't know if it's appropriate, but definitely for the company perspective was what changed the success of the company. So RNA vaccine, after the success of this COVID, could be eventually used for cancer. However, apparently they are less good for cancer, and I don't want to go into details why. So still, the peptide vaccine is still the best formulation that the scientific world agrees for cancer vaccine. So this is a little bit more of comment on the type of formulation that you want to use, what is covering, what we are doing now to expand this coverage of the population. Then there was other question interesting, if we try to combine the vaccine with different ALK inhibitors. We did it with crizotinib at the time, and then we jumped with lorlatinib. So now the new data are with lorlatinib. However, we are pretty sure that the vaccine could be efficiently combined with alectinib. We are almost sure. We simply couldn't do technical the experiment because all the experiments are time and money consuming, as you can say. I mean, this type of work requires really a lot of effort, economical and personal effort. But because it worked efficiently and very well with two different ALK inhibitors, we don't see a reason why it should not work basically with any ALK inhibitor. We did some small experiment to confirm that, and in fact, it's working with almost any ALK inhibitor, but technically we had a final proof only with crizotinib and lorlatinib. And then, of course, there are very interesting questions whether and how eventually combine the vaccine with other type of immunotherapy. And somebody noticed that our vaccine works quite well with immune checkpoint therapy, in particular with anti-CTLA-4 antibody. And maybe this is more is a very important question again, so a compliment to the audience. However, the complexity of combining immunotherapy and ALK inhibitor is really a clinical question that maybe I can turn to Mark, because of course in mice for some reason are very tolerant to a triple combination of an ALK inhibitor, an ALK vaccination, and immune checkpoint therapy. However, Mark can comment on how the clinical perspective can see these type of combinations. Yeah.
Right. So the trial itself is not open and activated yet. We are hoping to feverishly get it open and activated to start recruitment within the year. We are currently working on the FDA side to try to, since this is a novel product, a new product, we need to get clearance from the FDA to kind of submit a lot of documentation to explain what the therapy is, what the safety data are from the experiments, to get the green light to then move it towards the actual human clinical trial. So we are working quite hard to get this open as soon as we can, and our hope is within the year. I think somebody asked about industry partnership or a corporate partnership for funds, and that I think those pieces have fallen into place fortunately. I think for some of the drugs that are now approved, like alectinib, which is made by Genentech, or lorlatinib made by Pfizer, these are huge industry pharmaceutical companies that fortunately have tremendous resources to test dozens of drugs, kind of figure out which candidate is the best to move forward with, and pour a lot of money into opening global studies and opening trials at 100 sites across the world very, very quickly. Now, with our vaccine, this came essentially out of the work from Roberto Chiarle's lab. There wasn't a company that said, we're going to take this concept and idea and amplify and magnify it in this global scale from day one, which I think is, somebody asked me recently, which is a very smart and a good question. If we could discover COVID and develop a vaccine and roll it out to millions of people, in a 1-year time frame, why can't we do that with ALK? And I don't have a good answer to that question, except for people power and funding. But we are a dedicated but much smaller sort of homegrown effort here to take a brilliant idea with a lot of evidence supporting the strategy, and we're trying to kind of bring it to the clinic as much as we can, within some of the constraints that are somewhat out of our hands. But the purpose of the initial trial will be in phase 1. We of course want to make sure that, since this is a new product, that this is safe for patients. We also at the same time want to know if there's any activity or if it's effective. And so while we are assessing for safety and making sure that there are no unexpected side effects besides what you might normally expect for a vaccine, like a sore arm and maybe feeling tired or having some low-grade side effects for the next day or so, we want to make sure that there's no side effects beyond that. And so there are going to be 2 strategies. The first is to combine the vaccine with whatever kinase inhibitor a patient is already taking, where, as I mentioned, if the cancer had been growing on that kinase inhibitor, we would hope to add the vaccine to that and expect and want to see the cancer start to shrink again. The other cohort or strategy would be to combine the vaccine with a PD-1 immune checkpoint inhibitor. Many of you have heard of these drugs like pembrolizumab, which is Keytruda, or nivolumab, which is Opdivo, or atezolizumab, et cetera. So these are drugs that stimulate the body's immune system to attack cancer. And they can work quite well in some types of lung cancer, but we have learned that over the years that they tend not to work well for ALK positive lung cancer. And so the hope is that using the vaccine, we can trigger the immune system to recognize ALK and then use those approved PD-1 inhibitors to take the brakes off the immune system even further to attack the cancer. In the past, when ALK inhibitors have been combined with immunotherapies, or other such kinase inhibitors have been approved combined with immunotherapies. Some combinations are seemingly very safe, and others seem to create toxicities that are quite serious. So there had been some attempts to combine crizotinib with nivolumab, also called Opdivo, but that trial had to stop early because of hepatitis, meaning liver inflammation. Other combinations in other types of lung cancer had to be stopped early because of, for example, pneumonitis, which is lung inflammation. So we want to be careful initially and not throw too many drugs together at the same time, out of concern that there may be some toxicities that would then hamper the development of the vaccine if we have to put the brakes on the trial. So we're proceeding stepwise, in one cohort combining the kinase inhibitor with the vaccine, and in another cohort combining just the PD-1 inhibitor with vaccine, but without the kinase inhibitor, and try to work our way up from there as we learn along the way from these initial cohorts.
Terrific. So I did notice, obviously, you answered one question here, when will the trial start? And your hope is towards the end of the year. My gut feeling is that if you've got 24 people that you anticipate participating, that you would have a lot more people than that want to actually be participating. So how does that work? Is it just first in, first served, or how does that all work?
Yes, and that's the sort of heart-wrenching part of our job as an oncologist conducting clinical trials is we wish there was a spot for everyone who was interested at the time, but early phase trials have built-in pauses because again, these are brand new drugs, and we want to make sure before we, if we had 100 people, we wouldn't want to give 100 people the vaccine on, all on the same day in case there's some unexpected side effect. So we do these kind of staggered enrollments with 3 patients at a time, and then pause enrollment to make sure that after those patients get the vaccine, that there's no unexpected side effects, and then we're cleared to enroll the next cohort of patients. And so once we clear that period of time, that allows us to then expand to enroll at a little bit of a faster pace. And so initially, right, it would be kind of on a first come, first served basis for patients who are eligible, meaning that they're on an ALK inhibitor and they're experiencing disease progression, and otherwise their sort of overall health and laboratory values meet eligibility requirements, for example. So once the trial is open and enrolling, we will publicly post the eligibility criteria. This trial also will be initially HLA restricted, meaning that the HLA types will have to be determined, and only patients with specific HLA types for their immune systems would be enrolled in the first cohort.
Back from a search.
The trial...
Dr. Awad, did you say it was around about 50% would be covered under the, as far as the HLA types initially?
We think about 50 to 60%, which again, is a good fraction, but of course if a patient doesn't fall into that fraction, of course that's no good. And so, we are working to increase the number of ALK vaccine options and combinations for all patients. In terms of where the study would be offered or launched, because I'm seeing some questions here. So this would be, again, it's mainly due to funding. It would be in the Boston area. So it would be led at Dana-Farber, but we are extending and collaborating with Mass General so that they could open as a sub-site to the study. So that's quite limiting also, of course, because our patients are everywhere, but we have to start somewhere and unfortunately start small and then work our way up rather than launching a global study from day one. So hopefully as we learn more, we'll be able to expand not just the number of patients, but also the sites And I think, if things look promising, then with the right industry partner, I think that can also really fuel the expansion and development of trials in general. So we've been working with a smaller sized company here in the Cambridge, Boston area called Elycio Pharmaceutical Therapeutics, and they are kindly able to manufacture the vaccine. And so we have a manufacturing partner, but they aren't providing the funds to run and execute the trial for the vast, all the other kind of aspects for trial monitoring and scans and blood work and et cetera. So.
Terrific. So one of the questions I see here is, will the vaccine work in the brain?
Right, we don't know. We don't know. It is a great question, and so we also want to be cautious in terms of making sure that while somebody's getting the vaccine, that we don't want the cancer to start flaring in certain sites of the body. I think what we've seen or learned from other immunotherapy studies for other types of lung cancer is that, for example, some immune checkpoint inhibitors or PD-1 therapies, they can induce, like for melanoma, for lung cancer, they can shrink the cancer in the body, but also in the central nervous system or in the brain. And so we hope and think that immune cells can result in an intracranial or CNS central nervous system response, but we wouldn't know until we test it.
And obviously, as you have said many times here, and I think everyone understands, humans are not mice and mice are not humans. But what sort of durability of response did you see in your mouse experiments?
Yeah, so in the mouse experiment was extremely long. So when we vaccinate mice, we can generate this type of ALK-specific immunity that lasts at least 7 to 8 months, which is, you need to compare to the mouse lifespan. A mouse typically lives about 2 years, 1.5 to 2 years. So generating immunity for at least 8 months with one single vaccination is basically covering 30 to 40% of the mouse life, which in human equivalent could be some decades, at 20 years probably at least. Of course, if a mouse and human were perfectly identical. But the advantage of the vaccination is that if it works, you can repeat any time. So if your immune response over time tends to fade off, eventually you can go under another round of vaccination to boost the immune response again. But we definitely expect to be active for several years.
Terrific. That's very promising, and we hope that we respond as well as the mice do. Someone has asked a question here asking about ALK variants and whether in your preclinical studies, whether you did the experimentation on different variants.
Yeah, that's a very good question one more time. And there was also a very good question on mechanism of ALK resistance, whether the vaccine will be efficacious also against resistant tumor. For example, one example was through a MET alteration. So the expectation is yes. So the ALK variants should not affect the vaccination because the peptide that we identify, so the small portion of protein that we identify, are not those that are mutated, or which means change in structure during the development of ALK resistance to the inhibitors. So now the field knows very well what are the alteration of ALK that are associated to resistance. And to make the story short, none of these alteration will change the efficacy of the vaccine. So that's a very good news. Regarding the development of resistance through a different mechanism like MET alteration, but there are others that you know, again, the vaccine should not lose efficacy because the key point for the vaccine to work is that the ALK protein is still expressed by the tumor cells. And so when, for example, a lung cancer becomes resistant to an ALK inhibitor because MET starts to kick in and compensate for the ALK inhibition, that doesn't mean that ALK is not expressed anymore. ALK is still there, expressed, however, blocked by the ALK inhibitor, but the protein is still there. So the vaccines should still work against those mechanisms of resistance.
Terrific.
Maybe Colin, I'll just express some gratitude also, you know, as we're talking about resistance to ALK inhibitors. I think many folks here on the call tonight have heard about and even participated in what's known as the spacewalk study, which has also received a lot of philanthropic funding due to all the terrific folks involved in research. So just for those of you who may not be aware, in general, if a cancer has previously responded to an ALK inhibitor, but then starts to grow again, as many of you are well aware, one of the key first steps before deciding what the next therapy could be is to try to understand the mechanism of resistance to the prior ALK inhibitor. And that really, in some cases, can help us direct or you know, personalize what the best next line of therapy should be. So, for example, if there is another mutation in ALK, then it might make sense to switch to an ALK inhibitor, a different ALK inhibitor. If there's a new MET amplification, then considering a combination or a switch to therapy that targets MET, et cetera. So from our initial first cohort of patients who had participated in Spacewalk, and that's a way for patients anywhere in the country, here in the United States, who have developed resistance to an ALK inhibitor to get free blood next generation sequencing to try to determine within the plasma the mechanism of resistance. I can put a link to that study here in the chat. But we just published on the first cohort of 100 patients, of whom we had results on 60 at the time of the publication. That was published in Journal of Thoracic Oncology just recently. And so that study is still enrolling. If anyone has questions, feel free to reach out about that. But again, thank you to all the participants here and elsewhere for volunteering. We hope that it was useful. But as Roberto said, because the mechanisms of resistance to ALK inhibitors can be varied and somewhat complicated, our hope is that the vaccine would be somewhat independent of those resistance mechanisms. And even if there are different mutations within the ALK kinase domain or other genes becoming abnormal, the hope is that as long as the cancer still retains ALK expression, that the vaccine would still be hopefully effective. People have asked, you know, can the cancer develop resistance to the vaccine? Is this a cure? Are there, you know, these are superb questions, and we will not know, of course, until we start doing these studies. And you know, I think cancer is complicated, and cancer can develop resistance. We're trying to kind of sort of study that in advance in Roberto's lab as to how, you know, cancers may work around the immune system, for example. But our hope is that we will develop a vaccine that can overcome several different resistance mechanisms.
So I'm just going to take this opportunity to give a plug for the Spacewalk project. Dr. Awad is now managing or in charge of that project. And please feel free to correct anything that I say that's not quite correct, Dr. Awad. But there's two major reasons that I encourage people to participate. One is it can actually guide your next treatment. You get an analysis of kind of where your cancer's at. And based on that analysis, it can give an indication of whether you're better off moving to brigatinib or lorlatinib, or whether those things are not going to work at all and you need to go to some totally different type of treatment. So that's the one reason. And then the other reason, as you might have gathered by now, is that the results that Dr. Awad is able to garnish from that will help to develop further research. It helps to learn what the resistance mechanisms are, and based on that, it accelerates the development of new treatments for all of us. So would you agree with all of that, Dr. Awad?
Yes, thank you. Thank you so much, Colin. And yes, I think
and the criteria, my understanding is that you need to have progression on a second or third generation TKI. So any TKI except crizotinib, and you must be residing in the US. Unfortunately, they're hoping to expand it maybe to Canada at some point, but the international barriers are not all that easy to break down. So. A great question here from someone: do you need more support, money, awareness, etc., to boost the development trial of the vaccine or to accelerate it?
Roberto, do you want to start?
So that's, of course, is a question that is always the answer inside, that more money always helps. I mean, how can it be like that? But the reason, more specifically, why more money could eventually help is I thought we made clear that we are not stopping here. So this is just the beginning. And the trial will be rather small at the beginning. Of course, we have the support of a company. And if results will be good, we are pretty sure that the company will do a great job to attract more funding. But independently of this, this trial that will eventually start, we are already planning ahead. As Mark said, we are already trying to understand what will happen in the trial and what could be the reason why the vaccine could work less good as we hope, or if it works well, what are the way that we could even make it more potent or expand it to more patients, right? So this part, of course, is not covered by the company. And so we are doing ourselves in our research labs. So trying to understand what could be potentially the mechanism of resistance to a vaccine, like the mechanism of resistance to an ALK inhibitor, or if there would be better way to vaccinate the patients to improve the immune response, get a stronger response, get a response that lasts for longer time. And as I tried to mention before, how to expand this vaccine to cover not only 50 or 60% of the actual population, but eventually 100%, to be very inclusive and can be in the position to offer this vaccine to almost everybody. So this sort of development are done by ourselves internally in the lab. And since we strongly believe to this project, we are doing with the resources that we have. But of course, the speed of the development is limited by the funding. So as Mark mentioned, why the ALK vaccine is not going to the same speed of the COVID vaccine? Well, I don't have to tell you how many billions of dollars were invested in the COVID vaccination. And of course, the FDA completely changed the standard procedure for COVID. I mean, a typical vaccine goes under serious scrutiny, and you need to have very solid repeated experiment, phase 3 trial, very well controlled. For vaccine, all these rules, of course, were bypassed by the urgent need of the vaccination. So, however, everybody feel or hopes that that FDA will learn from this accelerated way to analyze the data and provide approval, and will apply similar approach also to cancer vaccine, not only to a vaccine against the virus, that of course is extremely important, but is not the only disease, right? And so that what makes approval of vaccine faster. I mean, of course, a lot of power, a lot of economical power, and of course the urgent need. And I think that the need is there for all of you. The need is definitely there. So we need to gather more economical power to speed up even more. Yeah.
And well, we can take this conversation offline further down the road, but there's a lot of talk about what's called an adaptive trial design. And is that something that you're contemplating doing? you contemplating working with the ALK Positive group to formulate how you're going to actually do the trial? Because we are encouraging people that run trials to really work with us so that, you know, the patient viewpoint gets taken into account.
Yes, absolutely, Colin. Thank you so much. And you know, we've received, you know, each step along the way, we've received great feedback, you know, from various grant applications and as part of the ALK Positive Longevity Grant, which is putting us sort of over the top to be able to open the trial. I know many patients serve on the board and are representatives to those grant agencies. And so we always take that feedback to heart and not only at those times, but as we're planning to launch the trial, we will share it in an earlier version with the ALK Positive longevity community and their funders to, you know, make sure that we really incorporate as much feedback as possible and still retain, you know, a very sort of rigorous assessment to the trial, so that, you know, at the end of the day, we will know, you know, the answer as to whether this vaccine is effective and should be expanded to a much larger patient population.
Wonderful. I do want to take this opportunity to let all the members know that Dr. Awad has also, on top of everything else, he's actually been one of the reviewers that helps to review our research selection process. And so on behalf of all of our members, everyone can put their hands together and really say thank you to Dr. Awad for doing that. That's very much appreciated.
Thank you. Yeah, Colin, I'll you know, I said this already, but, you know, I can't say it enough to thank everybody for, you know, all of their efforts in terms of focusing on research. I mean, I think, you know, everybody affected by cancer, you know, is going through so much already, you know, not just individually, but as a family and in their community. And, you know, to also dedicate time to research and asking questions and supporting and fundraising. I think it is so meaningful. Just, you know, for this vaccine trial, I'll mention, you know, Roberto and I applied for a grant to open the trial several years ago, and we were awarded that grant. It was $750,000 from the Koch Institute at MIT here. And unfortunately, that wasn't enough to open the trial, let alone to manufacture the vaccine, which costs in and of itself hundreds of thousands of dollars. And so they were very gracious with us. They said, you know, we understand running clinical trials costs money, so we will keep this sort of safe, this money safe for you until you can raise the rest of the money. We have since applied for NIH type grants and others. And, you know, there are some questions and skeptics out there. And so not everyone, you know, is as enthusiastic about, you know, different types of approaches. But, you know, I think what did put us over the top was the recent, you know, wonderful receipt of the ALK Positive Longevity clinical trial grant, which together with the earlier grant is what is enabling us to open the trial. So whereas we were hoping to have, you know, all the pieces in place, all the funding in place already, I think it's only through the research and the fundraising that has been done in large part because of the ALK Positive patient community that is fueling this trial. So, so a huge note of gratitude to everyone out there.
Terrific. There's a question here from Carla, who is actually a medical doctor herself, as well as being a patient. And the question is, what kind of vaccines, the ALK vaccine, DNA, RNA, mRNA, how will you assess the efficacy of the ALK vaccine? We use circulating tissue DNA, PET scans, tumor markers, other markers. That's the question.
Great. So yes, this vaccine is a peptide vaccine. Peptides mean small protein pieces, kind of like the influenza shot, or so it's not an mRNA or DNA vaccine. So it's protein or peptide based. And as part of the trial, the primary way to assess efficacy will be with scans. So getting a baseline scan, measuring spots of cancer, and then getting follow-up scans and hopefully seeing that those spots shrink. That has kind of been the fastest way for us as an oncology community to determine efficacy of a drug and to move it along through different phases of clinical trials to get it approved. Along with that, we will be measuring ctDNA, which is called circulating tumor DNA. So that means cancer DNA that might be floating around in the plasma or blood. And we will also be checking immune cells in the blood, or T cells, to hope to see that before the ALK vaccine versus after the ALK vaccine, we'll be seeing a boost in specific anti-ALK T cells. So I think the best way to see effectiveness is to see cancer shrinking, but secondary points to assess the efficacy will be measuring things in the blood as well.
If everything goes really well with the phase 1 trial, how long would you expect it would be before a phase 2 would start, and how many patients would you typically enroll in a phase 2?
So I think our hope is to accrue or enroll to the phase 1 within 1 year, and if we see some promise there, and if all along the way, even before the trial but after the trial, we will. We are already still trying to develop additional vaccines to span the spectrum of all the different HLA types in the patient population. And so I think the hope would be that if we're starting to see promising results and we can kind of create a snowball effect in terms of funding, manufacturing of the vaccine, et cetera, that we would then move into a phase 2 study of probably 50 to 60 patients.
And if it got to that stage, if it looks like it's going to prolong the effectiveness of an ALK inhibitor, would you expect that you might get some funding from the manufacturers of the ALK inhibitors, because it would allow patients to stay on their treatments for longer, and it would be in their interest to make sure that patients can stay on their treatments for as long as possible?
We would really hope so, yes.
Great. I'm going to do a completely non-vaccine related question here. There was a question here about sun sensitivity. And the question was, why is it that those on alectinib should not be in the sun? Is it direct sun, or is it the heat from the sun? I'm a sailor and need to know how to shield the sun.
Well, great question. I'll also open it to if there's somebody that wants to share their own experiences in terms of as a patient being on various therapies and sun sensitivity, because you all know this so much better than me. But I would say in general, even without being on therapies, just for skin cancer prevention, we generally advise and recommend avoiding a lot of direct sun exposure with hats, with sunscreen, et cetera. There are certain kinases, and everybody's different. Everybody has potential different side effects than others to the same drug. And so some people find that they don't have any skin or cutaneous side effects from drugs like alectinib or Lorlac or others, whereas some patients might find that their skin is more sensitive, or they might burn more easily, or feel some sensitivity to the skin with sun exposure. It's not clear why that happens, but for whatever reason, certain drugs, and this isn't unique to ALK inhibitors, certain drugs can cause bad rashes even without sun exposure. Certain non-cancer drugs, like certain antibiotics and things, can cause some skin sensitivity, particularly to the sun. So I think people often kind of learn what works for them, or what side effects they do or don't have, to kind of have a sense for that. So I see some people are mentioning here some of their experiences also, but so we think there may be some sun sensitivity. We don't know why, but.
Right. And there's some questions here that you've already answered, so you can just give us super quick, short answer to this without having to go into any detail, but there's a question here. Why not trial this ALK vaccine in patients who are currently no evidence of disease?
Yeah, thank you, Colin. I alluded to this just briefly in that that can be a difficult, that's a wonderful state. We want patients to be in no evidence and we want to keep them there. But with a new drug, if you're adding it at that time, how would you know if it's working? You would be looking for sort of a lack of progression. And if somebody has no evidence of disease, I think we wouldn't really be able to tell, is that ongoing state of no evidence of disease due to the vaccine or due to whatever got them there in the first place. So the initial trial would be for patients whose cancer is apparent on a scan and is progressing or growing. I can envision then, if that looks successful, we would want to then move the vaccine up, potentially into the first line setting, combining it with a first line ALK inhibitor versus the ALK inhibitor alone, and hopefully seeing an extension of benefit compared to just using an ALK kinase inhibitor alone.
Yeah, if I can add a little bit comment on this, more on the research side, Mark is very well said, that vaccine against tumor historically work best when the tumor is small. So it's really a battle between the immune system, how many cells the immune system can arm against the tumor, and of course, how many cells of the tumor are there. It's like two armies battling each other. So of course, the smallest is the tumor army, the better chances are for the immune system to overcome and completely kill them and eradicate them. So as Mark said, for clinical reasons, we need to do the clinical trial in a different setting. But in the future, the ideal situation will be to vaccinate patients when they are really at the minimal evidence of residual tumor. Even if the tumor is not seen anymore by CT scan or other type of imaging, possibly there are still some few cells there that could eventually come back after even several years. And we want really to kill them all. And that's what is the immune system is doing best, really when the tumor is small. So that will be eventually the ideal setting.
And correct me if I'm wrong here, but I think most of us here understand that the ALK TKIs basically kind of put the cancer to sleep, but they don't actually totally get rid of it. Again, correct me if I'm wrong, but my understanding is that if the vaccine turns out to be phenomenally successful, it can actually be curative.
Yeah, that's exactly what we see in mice, Colin, that the ALK inhibitor can control the tumor, put the tumor asleep, but not cure the mice completely. In contrast, the ALK inhibitor and the vaccination can cure mice because the vaccine does what the ALK inhibitor cannot do. So eradicate all the cells, even the very last cell that is there, and cure finally the mouse, and we hope also the patient.
Would there be a possibility of the vaccine actually working just on its own without a combination with TKIs or immunotherapy, other immunotherapies?
Yeah, that's again is a good question that relates to what basically I just said, that the ALK inhibitor are a very important tool to reduce the dimension and the activity of the tumor. So it is possible that the vaccine could work alone. However, such a wonderful tool like the ALK inhibitor is just to be exploited at the maximum possibility. So there is really no reason to go with a vaccine alone when these inhibitors are quite well tolerated. So the ultimate idea will be to reduce the time on which patients are on an ALK inhibitor. So if we reach the scenario in which the ALK inhibitor can greatly reduce the tumor mass and the vaccine can eradicate the final tumor cells, eventually patient could stop taking the ALK inhibitor and being cured. So that's what we hope, that at least in a subset of patients this will happen. Yeah.
Wonderful. Now, I love this question because it combines the spacewalk questions with the vaccine questions. Are you using the blood work from the spacewalk trial participants to ID patients with the correct HLA types?
That would be great, Colin. The funding for so HLA typing has separate costs compared to doing the DNA next generation sequencing. So the initial intent of the spacewalk study did not include HLA typing. And so that's not being done as part of the spacewalk study, although it's a great idea. We are offering it to patients who are coming through our clinics here, and I know that's also being considered at other sites as well. So, so it's not built into the spacewalk study, but that's a great idea.
All right, Emily, do you see any questions that you want to ask here?
Well, there was one that was actually not, it's vaccine related, but not ALK vaccine related. And someone was asking, with the recent EUA of the J&J vaccine, do you recommend that cancer patients try to get a particular COVID vaccine, or is any of them effective for, will any of them be effective for lung cancer patients?
This is a great question, and you know, it's somewhat outside you know, my expertise and field. So I would say that the distribution of the vaccine is so variable and so unpredictable and varies state to state and center to center, and we have been lobbying very hard for patients with lung cancer to be prioritized ahead of, you know, some other scenarios to get access to the vaccine, but every state is handling it differently. And so, you know, here in Massachusetts, for example, the first wave was just people over age 75, and then they moved it to people over 65, and then with people, you know, with certain comorbidities, two comorbidities, cancer is one, but you need a second. So, you know, there are all sorts of limitations and barriers, I think. And then even if somebody meets all these criteria, the supply is just very, very limited. And so I'm sure, you know, there are dozens of stories across, you know, the participants here tonight of people, you know, I've known people who have signed up, who, you know, got a slot miraculously, and then it was canceled, or they didn't have access, or, you know, they heard that it was going to be available through their grocery stores or pharmacies or wherever, and then, you know, the slots were just scooped up with it. You know, it's kind of like, you know, tickets to a big, you know, performance or something like that, where it's so hard to come by tickets. But so I would say that the more people who get vaccinated in general, the better. And so our hope is that through all of these vaccines, Moderna, Pfizer, J&J, and others, that that the number of new COVID cases will go down and down. And so I would say that if somebody has the opportunity to get a vaccine, I would probably just jump at it. Typically, what I've learned so far is that, you know, me, myself, you know, we were fortunate to get the vaccine as a healthcare worker, but we didn't have a say or have a choice to say, I want this one or that one. And I don't know of anyone else who has had that, unless somebody knows otherwise on the call. You know, usually just get a slot, you show up, and they tell you what they have that day, and they give it to you and tell you when to come back. So so I think it seems like the J&J shot is, you know, very effective, and you know, I think we'll also need to see, as those data are actually sort of released and published officially, to kind of look in a little bit more detail about it in comparison to the other ones. But I would generally say, you know, if there's an opportunity to go for it, just jump on it.
Great advice, great advice. There's a question here from someone called Gina Hollenbeck. I don't know whether you've heard of her or not. It says, could this trial be used to study ALK-driven cancer that is not lung cancer?
Yes, it's a great question. I'll turn it to Roberto, but I think the short answer is yes, it can be used in ALK positive cancers besides lung, but hopefully also extended to other lung cancers that are driven by genes other than ALK. Somebody earlier was asking about something like ROS1 or others, but Roberto, you've been doing some of this work as well.
Actually, as I briefly mentioned in introduction, the original work was done on ALK positive lymphoma rather than lung cancer, simply because at the time was not discovered yet. And it worked extremely well also in lymphoma, to some extent even better. The reason being that lymphoma is a single cell disease, does not really form tumor mass like in the lung, but is simply single cells going around, so they are more easy to be tackled by the immune system. So in lymphoma, definitely would be a great solution. Of course, a clinical trial in lymphoma will be more difficult because the patient are typically children, and so it's always more complicated to get an approval from FDA. So that's why we think that if we get some positive result in the lung space, it will be immediately translated to other type of ALK-driven tumor. And neuroblastoma is another type of pediatric tumor that is very, very frequently driven by ALK. But there are also a small fraction of colon cancer, breast cancer, some type of sarcoma, few percentage, but still all of them, they have very similar alteration like the ML4R translocation that is found in ALK tumor. So we project that this vaccine could work in any ALK positive tumor, actually.
Wonderful. I love this question. You can give a one-word answer. Are you accepting additional funding?
Yeah, we will be.
The answer is a big yes.
Big yes, and actually with a big smile, because we really need this type of support. I mean, the longevity and the ALK positive community has been exceptionally supportive for us. But I mean, any more funding, we will be used the best way that we can to work even harder and move even faster in this direction.
And I'll let you know that we're just starting to work on a new project where we're trying to connect researchers and capital venture and biotech together. So we'll certainly keep you front and center in our minds when we're doing that. Do you see any more questions, Emily?
One just showed up. Someone would like to know, how easy do you think it will be if the first trials are effective to then expand it to other HLA types?
Yeah, I can briefly answer. We think it will be fast, because we learned so many things to reach this point that if we are successful, we can learn from our experience and compress the 6 to 7 year work in much faster time. So we think that we could jump on other HLA types, especially if we get already the approval from FDA for this HLA. Typically, FDA has difficulty to provide the very first approval, and then once you get that, to expand to other is much, much easier. So everything should be much faster.
Terrific. That's really good. There was mention by Dr. Awad earlier about doing the ALK vaccine with immunotherapy. Can you explain under what circumstances you would be doing that?
So that would be an additional cohort to explore on the trial, Colin. So if somebody has been on different ALK kinase inhibitors before, but has had disease progression through them, and if somebody has also been on chemotherapy before, but needs a novel therapy, then it's in that setting that patients can enroll to that cohort to get the vaccine plus the PD-1.
So it'll basically be when everything else has been tried and is not working anymore.
Mm-hmm. That's right.
Okay. I'll just say, I'll read this out. It's not a question, but it says, thank you so much for all your work and interest in ALK-related cancers.
Thank you. Of course, it's a pleasure.
Do you see any more questions there, Emily?
No, I don't think so. And thank you so much for your time. And it's
Yes.
probably about time we let you get back to your Sunday night at home or at the lab.
I think Roberto has a microscope he needs to go to. So,
My Sunday work.
but I just want to say how fantastic it was. First of all, that you came in on a Sunday, and second of all, you give some hope, and third of all, where is the sign-up sheet? I might have to sign up. But just one last question that I have is, if you lorlatinib is the last TKI typically in your arsenal, would that be a person that you would be looking at as someone who's progressed on lorlatinib? Would be like a number one type of a position, or let me know what your thoughts are.
Yeah, thank you so much, Mark. We're trying to build in some flexibility to the trial such that a patient could come in, for example, if they're on first line ALK inhibitor, such as alectinib or others, and are starting to see the cancer grow on scans, but not to a point where they immediately need to switch to another therapy. That would be a nice point to try to add the vaccine to see if we can turn things around at that juncture. Or for somebody who's on lorlatinib, similarly, where maybe on scans we're starting to see the cancer grow, but again, not to a point where it's creating symptoms that require a more immediate switch to another therapy like chemotherapy or something like that. So we're trying to build in some flexibility there to allow patients on drugs like lorlatinib and others.
I'm going to throw out one last question. I think we all understand that lorlatinib is likely to receive first-line approval in the not very distant future. As a practitioner, would you, once it is approved as first-line, would you personally be prescribing lorlatinib or alectinib as the first-line drug?
This is a great question, and one that's sort of subject to debate, I think, among oncologists and otherwise. So there were two first-line trials to kind of mention. One is the ALK, called the ALEX study, and that compared using alectinib first-line versus crizotinib. And alectinib outperformed crizotinib quite a bit, and that led to alectinib's first-line approval. The ideal study, to kind of answer your specific question, Colin, would say a trial comparing the initial first-line use of lorlatinib to alectinib. But that trial hasn't been done yet. And so the trial that may lead to lorlatinib's first-line approval is the CROWN study published recently. And that compared lorlatinib to crizotinib. Now, we know that crizotinib is not the best first-line therapy anymore. We know that lorlatinib outperformed crizotinib in that study. We did not enroll to that study in the US because we already had alectinib's approval in the first-line setting. So it would not have been considered ethical to enroll and randomize a patient in the US, at least, where we already had another option for patients that was better than crizotinib. So this is a little bit of what we call a data-free zone, where we don't know if it's better to start with lorlatinib or alectinib. I think you also have to consider the side effect profiles of each drug and the dose of each drug. So the FDA recommended starting dose of lorlatinib is 100 milligrams, but that can be a high dose for many patients, and many patients have a difficult time tolerating that dose and may have a better time tolerating the dose of 75 milligrams or even 50 milligrams. And we know that lorlatinib can be very, very effective at much lower doses than the maximal dose as well. So I've heard some people discuss that they might prefer to start with lorlatinib, but at 50 milligram dose, for example, right off the bat. But again, we don't have data on that approach either, because that's not how the trial was designed. So this is where the oncologist's kind of personal preferences and experience may come into play. We try as physicians, as oncologists, not to make hard and fast decisions in the absence of data, but sometimes we have to use our sort of what we feel like is best based on the available but limited data. So I don't know the answer to your question in short, Colin. I think there is debate.
Yeah, and it's going to be very challenging too, because even if you had a comparison directly between lorlatinib and alectinib, and you found out that lorlatinib worked for, say, 9 months longer as a median than alectinib, it still leaves the question. We know that you can be on alectinib and then go to lorlatinib, and we've got many members here that did exactly that, and they've been on lorlatinib after alectinib for 1, 2, 3, 4 years. And so, even if you've got lorlatinib as being superior in the first line setting, it doesn't necessarily mean that it's best overall. So it'll be interesting to see where that all goes. So anyway, I want to thank both Roberto and Mark so much. You've been absolutely extremely informative. I think everyone's going to leave this call far more full of hope than when they arrived. And also not only more full of hope, but more full of knowledge and information as well. So thank you so much.
Thank you very much, Colin, to you and to all the organization, Erica, Jean, Mark, everybody, and of course, all the supporters and all the audience.
You
Thank
guys
you
want to
everyone so much. Thank you.
do you want to take yourselves off mute?
Yeah,
And you
thank
guys
you.
can
Thanks
thank
a
the
lot.
doctors.
Thank you so much.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you so much.
Thank
guys.
you.
Thank you so much.
Thank
Go
you.
research.
Yeah, go
Thank you.
Funding research.
Peace, Stephanie.
Thank you guys so much.
Thank you.
Thank you, Emily.
Cheers.
I'm going to keep the Zoom mic open for a while, the Zoom chat. So anyone who wants
guys-
to stay on board, stay on board.
Yeah, you're
I'll be
welcome.
here.
You can leave and go back to your families. if you
But I
like.
will
Thank
stop
you
the
so much
recording, so
for that.
nothing will be recorded.